Hi, my name's Rebecca Ahrens-Nicklas, and I want to thank the organizers for giving me the opportunity to come speak today. I'm sorry I can't be there in person, but I'm excited to share some of our work with you. So today I'm going to be talking about our efforts to develop platform gene editing therapies for rare metabolic disorders. And by way of introduction, I'm a physician scientist at CHOP. I'm a metabolic physician and a researcher. And I'll talk about our efforts that we've been trying to build over the past several years to try to attack and try to find an answer for some rare diseases. Here are my disclosures. So I don't think I need to convince this audience of why thinking about rare metabolic disorders is really important. But sometimes I speak to groups where they just aren't aware of the huge unmet need that our populations and our patient populations face. We all know that while individually rare, collectively they're actually quite common, and many, if not most, lead to neurologic dysfunction, and several lead to early mortality, unfortunately. But there actually has been a lot of interest from industry partners and drug developers and academics trying to do this type of work because there really can be a logical translational path in our rare metabolic disorders space. And I think it's important for us to remember that we have to convince others of this. So first, we know the molecular cause, so the specific genetic change that causes disease for most of our patients. And we often know that many times it's amenable to replacing or correcting a gene or misspelling in a gene. And so we have a logical way to attack the problem. Importantly, also in metabolic diseases, including CDGs, many have established clinically relevant biomarkers, things that we measure regularly. And we also know that platform technologies can be employed for many different related diagnoses. So essentially, you would just adapt a technology to meet a new disease. So many years ago, I started collaborating with my close friend and colleague, Kieran Musunuru, shown here, to think about how we could develop liver-directed base editing. Now, Kieran's an adult cardiologist. He really did not think about metabolic diseases at all until we started collaborating, but he had been using this tool to try to attack a different problem, elevated cholesterol levels. Essentially, the idea there would be that for patients with severe cardiovascular disease, you could edit or change the spelling of a gene that causes that leads to the synthesis of cholesterol or other lipids, and you could maybe turn off those genes in the liver. Now, when we started collaborating, it became clear that you could essentially retool or recalibrate those exact same types of drugs to now try to correct genetic misspellings in the liver in the case of our patients with metabolic disease. And all these drugs that I'm going to speak about today are essentially this fat bubble here, something called a lipid nanoparticle shown in blue, with two different RNA molecules in them. One is a messenger RNA. It encodes what you can think of as the pencil or eraser, the thing that actually goes and changes the genetic variant that you're trying to correct. And then a blue squiggly line here shown is the guide RNA, and you can think of that as the GPS signal telling the editor where to go. So there's a couple different types of editing. These are just showing two of them that were designed and developed by David Liu and his group that essentially allow you to make different types of corrective edits in the genome. The first that we're going to speak about today is base editing. Again, you can think of that as a pencil and eraser that allows you to correct certain types of genetic misspellings, but not all genetic misspellings. And then prime editing, which you can think more of a word processor that allows you to change a variety of different types of genetic variants, a larger selection than base editing. The work we've been focusing on first has been on base editing because technically it's much easier to deliver into patients. So that's what I'll be focusing on today. When we started working together several years ago now, we decided to pick a disorder where we thought we could start clinical trials in an adult population. In our case, we chose PKU or phenylketonuria because it's a relatively common rare disease, and there are recurrent variants or recurrent genetic changes that we thought occurred in enough living adults that we could essentially, like I said, eventually enroll into a clinical trial. And I'm going to shorten years of work into a single slide. But in the case of PKU, the problem is that you can't break down phenylalanine, which is an amino acid from your diet. And when that phenylalanine builds up, it is toxic to the brain. So after a lot of work, we essentially tweaked that combination of the GPS, that guide RNA telling the editor where to go, and the specific editor to develop a candidate drug to correct the genetic misspelling that causes the most common cause of PKU. And this is just showing in a mouse model of PKU at the bottom, mice that are untreated in green and orange have very, very high levels of phenylalanine, similar to my patients. You then give them a one-time dose of this gene editing drug, and within 48 hours, the phenylalanine levels entirely normalized. It also normalized in the brain and improved functional behavioral phenotypes in this mouse model. So when I saw these data, I thought, wow, we really need to think about how we could adapt this to more patients with metabolic disease and really advance this for the kids who have the most severe variants. And so we decided to transition, in addition to continuing to work on our PKU work, I should say, we decided to add on thinking about urea cycle disorders. You may ask, why did we pick urea cycle disorders? There's a variety of reasons. One is there's a huge unmet need. So patients with urea cycle disorders cannot break down the protein in their diet into urea. And when that happens, there's a buildup of a molecule called ammonia, which is toxic to the brain. And unfortunately, if left unchecked, can kill infants or adults or children very, very quickly. And there are a number of practical reasons why we started thinking about urea cycle disorders beyond just the fact that it's a very severe group of diseases. First off, we know that liver transplant can actually make a huge difference biochemically in the lives of these patients. Unfortunately, severe urea cycle disorder patients often present as neonates in the first couple days of life. And so during that time period where they're waiting to get a liver transplant, they can have real neurologic injury and significant risk of death. And so we thought that by correcting the liver, essentially trying to give a molecular version of a liver transplant more quickly than it typically took for a child to grow big enough for a liver transplant, we would have the possibility of maybe changing the trajectory of the disease. The other reason, and this is very practical, and I think it's important for rare disease communities to hear this, is that right now, that lipid nanoparticle, that soap bubble that exists, the ones that have been tried in clinical trials, essentially all go to the liver. The liver in our body is important for detoxifying things from the blood and just sort of sucks up anything you give into the bloodstream. And so while we're really good at delivering gene editing tools to the liver, we're not very good at delivering them to other places yet. Things like the brain or the muscle are much more difficult to reach. I think it's important that we will get there. And I think that there's a lot of great scientists working to develop better delivery technologies. But for this first use case where we were trying to develop a personalized editing therapy, we had to pick a disease where if we fix the liver, we had a chance of improving the outcome. And so that's a big reason why we chose urea cycle disorders. So that PKU work I showed you took about 2 years to finish. And we knew that for a patient that's born with a urea cycle disorder, we didn't have 2 years to essentially pick the perfect combination of pencil and eraser and guide RNAs. Like we did not have that type of time. And the reason being, these are very severe diseases. 50% of infants with severe CPS1 die in the neonatal period. And as I mentioned, liver transplant is a standard of care, but patients are at very high risk. And when I mentioned CPS1 here, I should say that is the first step in the urea cycle. We chose to work on this disease first, mostly because a patient came to us or we became aware of a patient and there was an unmet need. But it could have been any other urea cycle disorder gene that, as long as we rationally could come up with a way to develop an approach to a corrective editing therapy. So how do we cut down the time? So I told you in PKU, it took a very long time to sort of make the perfect editing solution, but we had to come up with a faster way. And we did a lot of things in the lab, including putting multivariants into cells and sort of streamlining how fast we optimize that solution. So that combination of editor and guide RNA in cells. And we did the same thing in mice. So I don't know if anyone in the audience has ever been involved in developing a mouse model, but it takes a long time and you don't always actually get the mouse model that you want to develop. And we needed to have a mouse model that had the human sequence that we were trying to edit, because that GPS signal, that guide RNA, is specific to the human mouse, or is it specific to the human sequence, not the mouse sequence. So we wanted to be able to test it in mice. So we came up with some ways to essentially introduce human variants into mice in order to be able to measure editing. So with these tools, with these sort of ideas and ways of working in the lab, we could validate editing solutions much more quickly. So we practiced. We took variants from the literature, we took variants from patients we had known previously, and just worked to see how quickly we could go from identification of the genetic cause of a disease to a potential editing solution. And Initially, it took us 18 months. And then finally, with a lot of practice, we got the timeline down such that we thought we might be able to try to do this for the first time. And right around that time, a baby was born. And I feel odd using his name, but his family has given us permission to do so. And I think many of you have seen pictures of him in the media. But he followed the course of a very severe urea cycle disorder patient. Around day of life 2, he became lethargic and critically ill. His ammonia was found to be higher than 1,000, which is incredibly high, and he was started on life-saving dialysis. And a lot of media attention, a lot of stories have sort of drawn attention to how good he looks, how well he is doing. I will tell you, while all this gene editing stuff is exciting, the reason why he's doing as well as he is because a good metabolic physician, a good ICU attending realized something was wrong and got him the care that he needed very quickly. And so I want to make sure that we recognize that good metabolic care, good neonatal care is essential. Our metabolic lab confirmed his diagnosis very quickly. And then within a week, through rapid genome sequencing, we knew the specific genetic misspellings that caused his disease. So it was at this point that we started thinking about going into the lab and using that pathway, using that expedited way of identifying a gene editing solution to try and see if we could come up with a solution for these variants. I should say, while this was going on, he continued to get gold standard clinical care. So after he was on dialysis and stabilized, he was transitioned to chronic urea cycle disorder management, including a protein-restricted diet, nitrogen scavenger medications, and then the plan was actually to remain admitted to the hospital for close monitoring while he grew large enough for a liver transplant. And so that's what the family chose to do. And they did an amazing job caring for him and their family throughout this process. So just because this research path was going on in the laboratory did not mean that his clinical care changed at all. In fact, he was actually listed for liver transplant around 5 months of age, much earlier than any other patient we had because he had a such severe form of this disease. And if he had received a liver transplant, that would have been wonderful for his family. But it turns out that the path I'm going to show you of developing a personalized editing therapy became an option before that, and the family opted for that path. Okay, so in the lab, a really talented graduate student named Sarah Grandinetti did all the work to essentially identify the right editing solution for this specific case. So she made cell lines, she put the variants in cells, she screened a variety of combinations of the editor, the mRNA that encodes the editor, and that guide RNA or GPS, because you have to get the right combination to make sure you optimize editing. And it was only when we had a solution in hand in a dish that we thought maybe we should try to move this forward and think about this from a clinical perspective. And it was at that point, with the consultation of our medical ethics teams with our IRB, that I had the first discussion with the family. This had all been done in a de-identified way. No one knew who this child was. We were just working off of a variant. And it was really an amazing conversation the first time I approached them and started discussing the possibility of research. I told them I have no idea if this would ever work. I have no idea if we would even be able to manufacture a drug. I had, before approaching them, come up with a blueprint, come up with a group of collaborators, essentially a way that we could manufacture the drug, going from cells in a dish to a first in human therapy, or experimental therapy, I should say, is not a straight roadmap. But I had at least assembled a team before I approached the family, where I thought we might have a path forward. But I was honest with them that I had no idea what roadblocks would come up along that path. And I stressed the importance of continuing to receive standard clinical care throughout this whole process. And I really just wanted to ask them, did they have any interest in the research continuing on the laboratory side? There was no way that anybody was ready to consent to an experimental therapeutic at that point in time, but I wanted to start the conversation about whether or not they were interested at all. And from that first discussion, they were amazing, essentially very quickly pivoting from what this meant for their child to what this meant for the rare disease community. And they gave us permission to go forward in terms of studying the therapy and trying to work on this in the lab. And from that moment on, they were full partners in this. So we generated a patient-specific mouse model by the time he was a little over 2 months old. And then we started doing the process of what's called an off-target nomination. So remember, I told you, you want that pencil and eraser to go change the genetic misspelling at the one specific point in the genome. You don't want it to go do other things and make changes other places. And so essentially, you do a very robust process of trying to predict where it might go, what might happen, and then looking for that in cells. We then had a meeting with the Food and Drug Administration before submission of what's called our investigational new drug application. This is really important. They were incredibly supportive and gave us guidance about how best to optimize safety and how best to move this forward. With our plan in place after discussing with the FDA, we did what's called a toxicology study or a biodistribution and toxicology study in non-human primates. This specific lipid nanoparticle had been in humans before, in adults, but I thought before we made essentially a version for a child, it was really important that we do a robust toxicology study to see what could go wrong. And what we saw was predicted essentially before we even started by the type of drug it is, which is elevations in liver enzymes. So lipid nanoparticles can absolutely cause elevations in AST and ALT. The non-human primates had no other signs of liver dysfunction. And it's really important that we know the risks so that we can help discuss this with the family. And so this was part of the consent process going in to the treatment, to the experimental treatment. We tested the drug in animals. We didn't have very many mice from our original colony, but we were able to show what's called a dose response. Essentially, you increase the dose of the drug, and we saw more editing. We then finally got his clinical batch and tested it in cells and showed that it had a nice dose-dependent editing effect. We finalized our off-target analysis with that clinical batch. We submitted what's called a single patient expanded access IND of the drug to the FDA and received clearance to release the drug and dose the drug about a week later. He then received 3 doses. This is showing 2 of them here. Okay, so I want to highlight again, this is a single patient expanded access investigational new drug application. What do all those words mean? Essentially, this is part of clinical care, and there's very strict guidelines about what type of indications, what type of diseases, what type of drugs could be used through this pathway, and he met those qualifications. Really, it's to test an experimental therapy in a patient with very few options or no options as part of clinical care. This is what we actually did. We started with a very low dose to ensure safety. LNP drug products can be redosed. There might be side effects of that. That has to be explored further, but the thought was that we would start low and go slow. He could have a maximum of 3 doses. All the doses had to be given within 120 days. Mostly because this drug was only made for him, and these types of drugs aren't stable forever. And so we had to make sure that he got those drugs in a safe way, or those doses in a safe way, before the drug went bad. Also, we had to think a lot about his immune response. So essentially, he made no CPS1 protein, and we were worried if this worked and the gene suddenly turned on, that he might actually mount an immune response against his new working protein. And so we gave him an immunosuppression regimen. I'll be honest, this is something that we tried to do our best to estimate what he might need and to take ideas from the literature, but we all are figuring out how this might work. Importantly, the decisions to redose were not made by me. They were made by a clinical oversight committee, a multidisciplinary group of folks, including our liver transplant team, and others. And so when that decision to make, every time there was a possible dosing event, the decision to redose was made by this committee. This is what actually happened in terms of his dosing schedule. He received 3 doses, and thank goodness there were no serious adverse events. You'll see each dose escalated in its dose. What happened? Everybody wants to know essentially what happened, understandably. What I will say, this was given as part of clinical care. This was not given as part of a formal randomized controlled research study. So I can tell you what we observed, but I cannot necessarily make broad claims about how well it works or how safe it is. That has to be further evaluated in a formal clinical trial. But after dose 1, he could tolerate more protein. And after dose 2, his scavenger medication amount could be decreased. So we think it might have made his very severe disease more mild. Since our initial report, since the paper first came out, we've been looking at a number of biomarkers. One of them is something called blood urea nitrogen. So this is a measure that we measure in patients all the time. And what we saw was that, especially after the third dose, his blood urea nitrogen, which he normally could not make urea, was essentially undetectable. But after the third dose, it suddenly went into the normal range. So we do have some biochemical evidence that we think his disease is more mild. This is just showing you some of that data over time. So this is his BUN over time, and you can see that especially after that third dose, it went up. And another marker that we look at for safety called glutamine has remained in a good, safe range. He's currently tolerating more than the recommended daily allowance for protein. He's still on a low dose of one of his medications, his nitrogen scavenger medication, because we want to make sure that he's safe. We are not calling this a cure. We think that this probably made him more mild. He has been successfully weaned off his immunosuppression, and he's tolerated numerous infections without a metabolic decompensation, which is very reassuring. So like I said, he's probably going to continue to need some urea cycle disorder management, but early signs suggest that his disease might be less severe. It's possible to develop a personalized gene editing therapy in 6 months if the stars align, everyone drops everything, and nothing goes wrong. This is not typical. This is not how drug development can proceed if we're going to scale this. If we want to expand this type of work to other communities, to other patients, we have to come up with a system to do so. And I'll speak to that in a minute. We showed that repeat doses of an LNP-based editing therapy can be safely given to an infant, but we need more follow-up studies. We need other markers of how well these drugs are working, and we're working on that in our lab to launch in future clinical trials. I will tell you, we did not do a liver biopsy to directly measure how much editing happened in the liver. Our ethics board, and I agree, thought that the risk of the procedure outweighed any potential direct benefits to the patient. Like I said, delivery to other organs other than the liver remains a challenge, and ultimately we need to move from N of 1 studies to platform trials. So how do we actually scale this? We can do things in the lab. This is just showing you a number of different genetic variants where we can do all that screening in cells and figure out editing solutions. Doesn't work for every type of genetic variant. Like I said, base editing only works for certain types of what are called transition variants. But for a large number of variants, we can come up with an editing solution. How do you move those editing solutions from the dish to patients? So we've been working really hard to think about this since we started this project. And so essentially, if you think about that drug I showed you, most of the toxicity comes from that LNP formulation, so that lipid nanoparticle. The inside cargo, at least from an acute reaction standpoint, doesn't really change the risk. And so why do we have to go back essentially and do lots of large animal studies every time we want to change the drug to target a different variant? So we asked the FDA this, and they're in agreement that you don't need to, which is great. So essentially, the risk from taking version 1 of the drug, targeting, this person's variant, to version 2 of the drug, targeting a different person's variant, is what's called that off-target risk, right? So where the guide RNA goes. And you want to look for that risk in human cells. So we've worked out a plan essentially that once you understand the safety profile of the carrier, the lipid nanoparticle, to understand the safety of that RNA, that guide RNA, that GPS signal going to the wrong spot, we really should be doing that in human cells. And the FDA agrees, which makes the process much easier. Okay, so we talked a little bit about how you make the process easier in the lab. How do you look at this in clinical trials, right? Populating and standing up a rare disease clinical trial is hard, and paying for it is hard, and understanding safety signals in ultra rare populations is hard. So what we've convinced them, and I think it's the right way to move forward from a safety and an efficacy approach, is to set up what's called a master protocol study. So we've been joking around and calling it an umbrella of umbrella trials, but essentially you have one clinical trial where you can enroll patients with any one of 7 urea cycle disorders with any variant that might respond to this type of technology. And you can evaluate them all in one clinical trial so that you can sort of pool your safety data and really understand in a larger population whether these drugs are safe. And you can compare efficacy both within a specific gene and across genes. And this will allow us to really gain more information about if these drugs are safe and if they work. Okay, so that's the phase 1/2 clinical trial that we're thinking about and working on very hard. But as many in this audience probably know, moving from a phase 1/2 trial to a trial that gets you an approval is really hard. Getting drugs approved is hard in the rare disease space, but we have to do it because it's only once you get drugs approved that you can start seeking reimbursement. And that's really how you scale this to allow more individuals to have access. So we have been working with regulators to try to think about some novel regulatory pathways. And one that has come up, and we'll see how it works out, is something called the plausible mechanism framework or pathway, which essentially, if you have a clear molecular basis of disease, like you do in metabolic disorders, and you have a medical product that targets the biological alteration, for example, the genetic mutation, and you have well-characterized natural history or even a natural history in an individual, maybe you can use this pathway to essentially streamline the process of getting to an approval. It's interesting. I think the devil will be in the details. We are working on it. But I think what I will say is that if drugs are approved this way, it's going to be really important for sponsors, for physicians, to continue to collect safety data and efficacy data once the drug is approved. Specifically, I'm calling all of us who are thinking about using this, myself included, that it's going to be essential to prioritize safety and transparency. This is always true in the rare disease space. Every patient is precious. Every clinical trial participant is precious. But we have to be transparent about what we learn, especially around safety signals, if we're going to be trying to approve drugs after treating fewer patients. Okay, so I put this figure up just by ways of sort of a summary of what I just talked about, because I know it's confusing. So this first column is the pathway that we took for that first N of 1 case. It's called an expanded access IND. There are certain criteria shown here that you have to meet in order to be able to be eligible for this type of path. You do have to do key proof of concept and safety studies. You do early manufacturing processes, but importantly, it only generates clinical data for that patient. Typically, that data is not used to support a research program that can move to an approval. So it is important for that individual patient, but its effect on the community in terms of robust data that can be used to ask for approval for a drug is not there. And so very quickly after, an expanded access IND or it is important to move towards formal clinical trials that can move towards approval. And there's now two ways that we know of that we can do this. One is through a traditional investigational new drug application in the standard development pathway, where, multiple subjects will be in a single trial. You do a lot of preclinical proof of concept and safety studies. And then the early phase 1/2 data would generate data to support a phase 3 and then eventually an approval. Contrast that slightly with what's this plausible mechanism pathway, where the patients might be similar in their populations and the preclinical work is similar. But the difference really is before you start your clinical trial, you need your process for manufacturing your drug to be entirely locked with the goal that that one clinical trial might support an approval. So I'm happy to talk more about this. If you have questions, feel free to reach out. But this is our understanding of the ways that you move forward. Okay, as we think about scaling and paying for this, we also have to make the case for reimbursement. This is our thought process for urea cycle disorders, but I think it's important that rare disease communities start quantifying the cost of caring for patients. The physicians that care for them, we need to do this work so that when these drugs become available, we can understand and demonstrate not just the health benefit if they are shown to be safe and effective, but also, in all honesty, for payers, this type of data are important. And for urea cycle disorders, liver transplantation alone is $1 million, and so you can very quickly start making the cost benefit justification. And so with that, I'd like to thank so many people that were important for this work. You can see it was really a multidisciplinary team of academics, industry partners, the family, most importantly, our regulator colleagues. The NIH funded all of this work, and so I cannot thank enough the federally funded science program in our country. It's essential. And I hope that you guys have a wonderful meeting. And if anyone has questions, feel free to reach out. Thanks so much.